Good morning, and thank you for attending our DiscoverX hosted webinar today. I'm assuming you have been able to see the screen. Let me do one quick diagnostic. And I think we're in good shape. Okay. Sorry about the slight delay. You should be getting screen as video as well as audio. Please don't worry about taking notes from the slides. We will be sending you PDF versions of the slides as soon as the presentation concludes. If you have any questions, please enter them at any time in the Q&A or the chat box that's found in your WebEx bar, and we'll do our best to address those questions at the end of the presentation. Today, we will be talking about immuno-oncology drug development, profiling immunomodulatory biologics and compounds in human primary cell-based oncology models. My name is Sega Omina, and I'll be the moderator for this WebEx presentation today. And I'd also like to take this time to introduce a good friend and colleague, Dr. Allison Omani from our BioSeq team, and she is the Vice President of Research Biology. Sorry, she keeps on climbing up the ladder, and her title keeps changing. So, okay, without further ado, I'd like to have Allison take charge of this webinar.
Thank you, Sega, and good morning, everybody. I'm hoping that you can hear me. So, thanks for taking the time out to listen to our presentation today. We are certainly very excited by the data that we have accumulated in this space, and certainly it matches the excitement that currently exists in the clinic for this class of compounds, an area that I, both in my research and academic careers, focused on. So I'm familiar with this biology for a very long time. So personally, I'm really thrilled to see it come to fruition and make such a positive impact on so many patients' lives. This introductory slide is really positioned quite differently. I'm sure those of you who have been to any immuno-oncology presentation or seminar or manuscript or review in the last couple of years have seen some form of this Chen paper figure. And really what I wanted to use it for today was really as a graphic to illustrate that the relationship, the complex, convoluted relationship between a host immune system and the emerging cancers really has been known for a very long time, going back more than a century to the work of William Coley and others that demonstrated this really antagonistic battle going on between the host and the emerging tumors. And so this slide really represents an enormous body of work on the different aspects and components that we've garnered in really very elegant research over the last 150 years or so. However, it's only really in the last number of years working on some seminal data from Jim Allison and Carl June and others that we have begun to actually finally get a therapeutic handle on this biology. And really, I think that relates to the fact that it's been very difficult to translate the huge enormity and complexity of the data in these research models and transition or translate it into the classic approaches we use in drug discovery. It's really been difficult to mine it down to in vitro approaches that allow us to identify potentially active compounds. And then an even greater obstacle comes from taking this data and putting it into such a form that it's actionable and guiding into making better therapies that will end up being more efficacious and safe in the patients. So really, these are sort of three stages, but these arrows that I illustrate here in this image are enormous obstacles. So what we've done at DiscoverX is take a rather methodical approach to the drug discovery continuum in this immuno-oncology space. And really, we've reduced it to what at first appearance look like very basic and simple questions, but are nonetheless fundamentally important. And what we've tried to do is to take our expertise, to take our assays in our portfolio and help address and provide valuable information in answering these questions. So in blue arrows, we've sort of represented really at a very high level some of the fundamental stages along the drug discovery continuum. These apply to large molecules, small molecules, and combinations thereof. And so some of the fundamental questions that we as a broader team across DiscoverX's portfolio groups have come up with are really key questions. One, how do I screen for a functionally active lead molecule? Two, how do I know if the efficacy of that molecule is going to operate in the highly complex tumor microenvironment model, in the highly specialized tumor microenvironment model? And then, because we're not treating cancer tumors, we're treating patients with tumors, how do I know if my drug is going to have impact both with respect to efficacy and safety outside the tumor, the specialized tumor microenvironment? And finally, a very important strategy, and certainly has been almost the standard of care in oncology for several decades now, how does my compound work in the context of a combination, be it either a rationally designed combination or be it the application to onboard therapies, and classical chemotherapeutic agents would be a category of onboard therapies for these new emerging immuno-oncology agents. So as I said, at DiscoverX, we have a broad portfolio of both target-based or target-centric and phenotypic assays that really are designed to impact and inform on compound development through the drug discovery continuum. Our DiscoverX's sweet spot has always been on the development of highly specialized target-centric or target-focused assays. And here the depth of coverage of these target-based assays largely reflects the drug discovery pipeline globally, where GPCRs still and kinases make up an enormous amount of target-based approaches. Along the bottom of the slide is where our team comes in, and this is the BioSeq primary human cell phenotypic profiling assay called BioMAP that was acquired by DiscoverX in 2012. And our assays are designed to model human tissue and disease biology, and essentially, therefore, are target agnostic and span really a wide variety of therapeutic targets and strategies. So today I'm going to focus, as denoted by these stars, I'm going to focus on the target-based checkpoint assays in the PathHunter suite, and then I'll spend the majority of my webinar going over the BioMAP application to building human primary cell-based tissue microenvironment models for tumor biology, and that's really what the relevant space for immuno-oncology is today. So again, going back to our drug discovery continuum questions, here I show you where the PathHunter target-focused bioassays are important in the IONC space, particularly for biologics in this space, and then where the BioMAP broad panel or disease-focused panels have been used at different stages to evaluate compounds and combinations. So the first question, the first fundamentally important question, how do I screen for a functionally active lead molecule? Well, some of the challenges that we face in this screening approach are that the target-based assays really require specific cell-based assays for each target. You have to have the right target expressed and the right biology being modeled to be able to get a screening answer. We have to have this in human cells. We all know that animal models do not faithfully recapitulate human immunology, more so than many other systems. And so we do actually need the complex T-cell receptor and T-cell lineage biology in the human context. There is the reality of donor variability. Now, we address that in BioMAP in a different way, but for screening campaigns looking at cell-based assays, you do want to have a robust and reliable screening assay that every time you come along with an agent or a combination or batches of the same agent, you can get a robust and reproducible response. And then a lot of the assays in this space, in this immune signaling space, have longer assay times. We all know about the extended MLR type of assays that are used in immuno-oncology can take 6 days or more. I know many labs are doing them at a 10-day window, and that's a long assay window to get an up or down or a yes or no answer. And of course, the protocols and the diligence required for executing these assays is certainly complicated and challenging. So for the target-based PathHunter assays, DiscoverX has developed a number of approaches taking advantage of its proprietary enzyme fragment complementation technology, or EFC technology. The basis of it is shown here, in which there is an enzyme that has two fragments that when separated are catalytically inactive. And when brought into proximity with each other, where they are able to interact, they form a catalytically functional enzyme, which is capable of hydrolyzing a substrate to emit a chemiluminescent signal. So essentially, these two components can be used to tag interactive proteins and enable one to see a signal when the interaction occurs, and then an inhibition of that signal when a blocker or an interrupter or a signaling inhibitor is introduced into the equation. So where that has been applied in the IONC space is the development of PathHunter assays for various immune checkpoint receptor constructs. So here's a graphic illustration of the constructs and cells generated to read out PD-1 or PD-L1 mediated biology. So in this case, the PD-1 receptor is tagged with the enzyme, one of the enzyme components, and the other recruitment factor, the SHIP phosphatase, is tagged with the other. When these two, when the PD-1 receptor is engaged and activated, it becomes phosphorylated and then serves as a docking platform for the recruitment of the SHIP phosphatase. The two tagged components of the EFC enzyme are then brought into close proximity, where they interact and form a catalytically active enzyme, which breaks down the substrate in the reporter cell line, emitting, as I said, a chemiluminescent signal. Agents that interrupt this, so blockade antibodies that block the engagement of the PD-1 by its ligand, or downstream kinase signaling that block the phosphorylation or the recruitment of the SHIP phosphatase, will all disrupt the co-localization of the two components or the two fragments of the enzyme, resulting in an inhibition of the emitted signal in a classic reporter inhibitory curve. And here we see this data shown very nicely with the two approved clinical PD-1 antibodies, pembrolizumab or nivolumab, or as their brand name, Opdivo and Keytruda. So what we see is a really very nice and compelling and robust and reproducible inhibitory curve why these checkpoint blockade antibodies are interrupting the engagement of PD-1 by its cognitive ligand, and a disruption of the recruitment of the two components of the EFC, and a dose-dependent inhibition of the chemiluminescent signals. This assay is also amenable to small molecule inhibitors, and here's an example of two Circ kinase inhibitors that would act to block the phosphorylation of the PD-1 tail. Therefore, the SHP phosphatase would not have a docking platform, it would not get assembly of the EFC, and the enzymatic or catalytic function of that reporter would be inhibited. And so here we see again another classic inhibition curve. So to date, DiscoverX has developed a number of these PathHunter assays for the specific checkpoint receptor classes, shown here PD-1, VISTA, and TIM3, which are all in the immuno-inhibitory class of checkpoint receptors, as well as co-stimulatory class, including OX40, CD40, and HVM. The assays that have been developed with these reporter constructs and lines have an easy protocol with a rapid result, which is, of course, highly compelling for a screening campaign. And they support the development not only of biologics, but also of small molecules and potentially combinations where you have a synergistic or shift in the curve. And these assays enable researchers to screen confidently with a highly sensitive result. So this PathHunter technology, again, is target-centric and is really designed to have a rapid and robust screening approach that's suitable both in early discovery, lead ops, and indeed later on in batch-to-batch comparison of different, in particular in biologic samples. So now we're going to switch gears a little bit and go to a more physiologically complex but highly clinically relevant model system, and that's the BioMAP system. And this system really we're positioning as an approach to answer these questions in the efficacy and biomarker selection arena of the drug discovery continuum. So some of the challenges, and this is by no means an exhaustive list, but I think it's some of really the initial challenges to get going in this space. So in vitro assays, even the ones that we've just described, while they're very valuable for giving you a yes-no reporter answer, they don't recapitulate or model the complexity of the human tumor microenvironment. And it's always worth reminding audiences at this point that immuno-oncology is a space that really came out of the classic immune tolerance biology, where the tumor has exploited those normal tissue tolerance mechanisms to its own advantage. And so in the same way that we can't envisage a strategy where we inhibit globally or systemically inhibit tolerance, that has been one of the challenges. How do you confine this therapeutic impact to the selective tumor microenvironment and not harm the patient as a whole? Animal xenograft models are not amenable to screening campaigns, and there has also been some questions as to the reliability and the translation of the responses in animal models to human patients. Again, the mouse immune system and the human immune system are fundamentally different. And then there's been the reality that there are appropriate response biomarkers in oncology, and particularly in immune-oncology, remain highly sought, and the current response biomarker, patient survival, doesn't really translate into the drug discovery process. So we have to find better ways of capturing those clinical responses and bringing them back into the in vitro drug discovery process. So at BioMAP, what we have done is we have expanded our approach that involves a systems biology strategy to combining in vitro human biology models with robust and interrogating mathematical or bioinformatics approach. In essence, BioMAP platform enables one to put a mathematical handle on in vitro human biology data. So we start in this approach by using human primary cells. And we build those as tissue microenvironment models. They're not tumor, but tissue microenvironment, where the disease is complex, complicated, multi-component, and multicellular. No disease is driven by one cell type with one cytokine or one growth factor. So we build these complex co-culture models with a variety of different stimuli that we call BioMAP systems. And by the term system, I mean a particular set of primary cells, co-cultured or monocultured, a particular set of stimuli to recapitulate disease or inflammation or tissue remodeling or wound healing type biology, and a particular set of protein-based readouts. And the protein-based readouts are designed to not only be biologically relevant, for example, an anti-inflammatory readout, but they're also designed to be clinically relevant, i.e., the activities of this compound on these protein biomarkers make it suitable for an autoimmune indication or immuno-oncology indication or a fibrosis indication. Because the systems are fully human with respect to the cell types involved, they're amenable and indeed ideal for profiling or testing large and small molecules and combinations thereof. And this is really where the clinic is going in immuno-oncology. It is going down the combination approach. And in addition to building these compelling primary cell-based models, we have also developed a powerful suite of bioinformatics and data mining tools to extract the value from this in vitro human biology data. And the goal really is to provide back information to our clients to help them make better drugs to treat patients, which is something that we all in the end want to do better. Okay, so here is a schematic in which we have adapted our classic primary human cell disease models to incorporate a tumor influence. So in the BioMAP tumor microenvironment, as opposed to our tissue microenvironment models, what we have done is we have taken our normal tissue cell types. In this case, we have two different models, one of which uses fibroblasts as stromal players. The other uses HUVEX, or vascular endothelial cells, to model the vascular or angiogenic component of the microenvironment. We use PPMCs to model the immune component of the microenvironment. And then we have added a cell line. The reason we use a cell line is that we want these systems to be robust and reproducible and screening amenable. And so primary tumor explants are not reliable. They have drugs on board and they have variants. A cell line allows us to incorporate this tumor influence into what is essentially a microenvironment model. We don't use these systems to read out a lot of biology on the cancer cell line. That's not the goal of these systems. Rather, it's used to read the impact of the compounds in the tissue and tumor immune microenvironment biology under the influence of these cancer cell lines. We have two of these models for two different cell lines. The HD29 stromal system and the HD29 vascular system, and we have also built these models with the H1299 non-small cell lung cancer cell line. So we have a colorectal adenocarcinoma panel and a non-small cell lung cancer panel. The panel I refer to is a stromal immune microenvironment and a vascular immune microenvironment. The way we develop and utilize these systems is we co-culture the host tissue and tumor cell types and allow them to come to confluence. I might remind you that the human tissue cell types are primary, they're contact inhibited. They will form a confluent monolayer in conjunction with the cancer cells. And then we add the immune cells and immediately add the agent, be it, again, a large or a small molecule. Within about an hour of adding the agent, we stimulate the cells, and we stimulate them with a submitogenic level of T-cell receptor agonists to prime the immune cells, because the immune cells got to the tumor in the first place. So they did recognize the tumor as being a non-self site, but at the site or within the intratumoral space, they are subject to the immune inhibitory effects of the cancer. So what we've done is we've primed the T cells within this tumor model to look for the impact of compounds on this immune tumor microenvironment biology. Compounds and stimuli are then assayed and incubated in the system for 48 hours, followed by reading a number of protein-based readouts. Before I describe the readouts, I want to give a little background on the biology that we have demonstrated that we're modeling in these systems. Here is a rather straightforward histogram that nonetheless I think powerfully illustrates the cytokine response or activation response in this system. Along the bottom, we have the different individual and paired components of the system. And all the way over on the right, we have the final system, the STRO-HD29, which represents the fibroblasts, the immune cells, and the HD29 cancer cells. What you see here is in response to the T-cell receptor agonist stimulation, PBMCs will make an inflammatory cytokine, in this case interferon gamma. We see the same with TNF, IL-2, and other inflammatory cytokines. When you co-culture those PBMCs with fibroblasts, you actually see an enhanced inflammation response. And indeed, this is the biology that would be relevant for autoimmune disease, such as a rheumatoid arthritic synovial joint, where you have the fibroblasts actually contributing to the inflammation environment. But when we add the when we add the HD29 cells either to the PBMC cultures themselves or to the co-culture with the tissue stromal cells, we see a strong inhibition of this immune activation response. So this is a rather clear demonstration that the cell line in this tumor microenvironment model is mediating an immune suppressive or an immune inhibitory effect. And that is the arena in which immuno-oncology is acting. Another question that we often get that I wanted to put the data in immediately is that all of our cell types, both the tissue and the immune cell types, are pooled from multiple healthy donors. This allows us to determine population response and not become reliant on a single donor for our activity profile. And then, of course, we often get asked, if you pool PBMCs from multiple healthy donors and they're not HLA matched, are you getting MLR? And so this data shows with the blue arrows that when you compare pooled PBMC donors on the right-hand side versus individual PBMC donors, up to 6 of them on the left-hand side of the graph, in the absence of the exogenously provided T-cell receptor stimulus, you don't really get any immune activation. So there's very little MLR going on here at 48 hours, which is consistent with several other labs. It's too short a time frame. Importantly, whatever basal MLR is going on is not only going on in the compound-treated wells, it'll also be going on in the vehicle control wells. And that's what's used to identify the activity of compounds, as I'll show you in the next slide. But here I wanted to illustrate not only is the response masked, any contribution of MLR is massively masked by the activation but in the stimulated samples, shown here in red, but also there's the reproducibility of the response. If you look across the models in which we use PBMCs from a single donor, you see, in fact, there is tremendous variation in the response. This is simply not acceptable for a screening campaign. In other words, if you look at a normal donor 4 here, he or she looks almost immune compromised relative to donor 2, and yet these are all healthy donors. And so by pooling the PBMCs from multiple donors, a minimum of 3, but often more than 5 or 6 donors, you get a much more reproducible response. And now when you screen your compound now, 6 months from now, or compared to something you did a year ago, the CVs are tolerable for a screening campaign. Single donors are too variable for screening any compound. Now finally, the result of this is that you have a BioMAP profile in which the compound's effects are displayed as the relative expression level of protein biomarkers compared to the vehicle control on the same assay. So here we have the stromal HD29 that's been used to profile GSK's MEK inhibitor, trametinib, at sub-Cmax or subclinical exposure levels. What we see is that the vehicle control is represented along the 0 line here. That's the intra-assay vehicle control. And around that, we have a gray envelope. That's the 95% historical vehicle control envelope that essentially serves as a 95% confidence interval. So when a compound is active outside this envelope, we annotate it or label it as a statistically significant activity of the compound. What you see here is that trametinib strongly inhibits the immune response of the PBMCs in this host stromal microenvironment model. Now remember, I showed you that the immune response was already suppressed in these systems. So here it's driven even lower by this MEK inhibitor, consistent with the biology or the mechanistic biology of this compound. We also see effects of this agent on other markers, inflammation markers, including VCAM and IP10. So it is generally affecting inflammation, which we would expect a MEK inhibitor to do. It's also affecting a CCAM marker, which is largely expressed on the cancer cells. And it's also increasing collagen 3, which could be a matrix stabilizing associated biomarker activity for this compound. So now I'll show you some more data. Here is the profile of pembrolizumab, the anti-PD-1 or Keytruda, that we've profiled both in the STRO HD29 and BASC HD29 systems that make up the CRC panel. What we see is that at 4 concentrations tested in triplicate, we see a profile of activities for this agent consistent with immune restoration or immune recovery. And this is very important because there's a difference between an inflammatory effect and an immune recovery effect that we'll talk about when we get onto the further slide. So we see a pattern of activities and identify response biomarkers unique to the tumor microenvironment that are consistent with immune restoration, as we see also in clinical reports for this compound. We see a similar profile, a similar pattern of activities, so to speak, when we look at the nivolumab or Opdivo. So both Keytruda and Opdivo show a pattern of activities consistent with immune recovery in these human tumor microenvironment models. We've also looked at small molecules that are already approved for oncology indications and actually show an intriguing and interesting ability to also recover or restore immune function. So here is a profile in the CRC panel for Paclitaxel, which we were quite surprised initially to see that it had immune recovering pattern of activities. But in fact, when we looked at another microtubule stabilizer, completely different scaffold, a completely different compound, but with the same mechanistic impact, we also see a pattern of immune recovery. So microtubule stabilizers then can be qualified as having the potential for IONC efficacy, and indeed Paclitaxel is in clinical trials with an anti-LAG3 antibody. So here is a pattern of activities that we can use to qualify IONC potential. We have also been able to compare IONC potential between similarly acting small molecules and large molecules. Here's a profile in the STRO HD29 system of Erlotinib, which is an EGF receptor kinase inhibitor, a small molecule, again showing a pattern of immune recovery. And then in parallel, when we look at cetuximab, which is a large molecule targeting the same EGF receptor biology, we also see evidence that there's immune recovery function for both of these agents. So that we can use these systems to compare small and large molecules, but also we can use them to compare molecules for potential to be synergistic in a combination approach. And I will discuss that later on. We've also looked for clients, and this is a collaboration with MedImmune AstraZeneca, in which we profile their anti-CD73 antibody. And again, we see it's not identical to pembrolizumab, which makes sense, but it is overall a pattern of activities consistent with immune recovery in these tumor microenvironment models. So we have shown compellingly that in addition to inhibiting responses by classic MEK inhibitors and other small molecule inhibitors used in the clinic, we also have the capacity in these systems to capture immune recovery or immune restoration biology. However, a second question that we've also applied our tissue microenvironment models to is addresses these questions. So, is my drug or is my compound or combination having a biological impact outside this unique immune suppressed tumor intratumoral space? And so challenges for this is that it largely relies on a single cell type, which doesn't really capture the biology, or an organ type, which is challenging for screening. It does often have a bias for particular targets or pathways. The model systems are often quite simplistic in the single cell approach and often isolated biology. And there is limited testing of broad-scale human biology with a drug. Simply put, you cannot do these type of experiments in patients, period. So what we have is a series of observational data, and then we try to model that in our system. So to this end, we go back to our flagship and our original BioMAP phenotypic screening platform, the Diversity Plus platform, which are 12 primary human cell-based tissue and disease models. So there's no cancer cell line involved in any of these systems. But what we're capturing here really is the biology of the patient outside the tumor. So when we look in this panel at Paclitaxel, remember Paclitaxel along with epithelion D was one of the microtubule stabilizers that had an activity profile consistent with immune activation. Here we see that the action of Paclitaxel outside the tumor microenvironment is largely what it is. It's an anti-proliferative agent. And here in the pink arrows, I'm showing you that this agent inhibits the proliferation of endothelial cells, coronary artery smooth muscle cells, and fibroblasts. This is how it works. And this is how it works mechanistically. We also see, though, some inhibitory effect on immune function. So these are inflammation models. And in that case, we see Paclitaxel actually being anti-inflammatory, even though in the context of the tumor microenvironment, it was restoring these. So there's a very significant disconnect between non-tumor-related inflammation and intratumoral immune inhibition. And they're two really critically separate compartments. And the reason they're separate compartments is if you come in with an agent that systemically increases or enhances inflammation responses, then you have really the elements of what was classically called a cytokine storm. You cannot see increased cytokines in the periphery of a patient. It needs to be localized to the intratumoral space. It needs to be measured in tumor biopsies. And when you have tumor biopsies, they're rare, they're precious. What do you look for? You look for response biomarkers that you've already identified in vitro models such as BioMAP. I also showed you that Erlotinib, or the EGF receptor kinase inhibitors, Tarceva and Iressa are their brand names in the US. This is a small molecule kinase inhibitor that had a pattern of activities, again, consistent with immune recovery or restoration. But here in this non-cancer tissue model platform, most of its activities are actually tissue remodeling. This is a lung cancer standard of care, and in bronchial epithelial system modeling lung epithelial biology, it's really impacting the matrix and the stability of this tissue. And so here we see activities that relate to efficacy, but also activities that are going to disrupt the lung function in patients, which is noted. Not only do we see that, do we see impacts in the lung model, but in the wound healing model we have here, we see an increase at EGF receptor levels. And that's a well-known compensatory response to EGF receptor kinases. They make more receptor in a way to compensate for the inhibition of signaling. And actually, compounds that have this signature in the BioMAP system are associated with predictive skin rash outcomes in patients. And in fact, the skin rash in patients on Tarceva and Iressa is used to determine if the patients are responding. If the patient doesn't manifest a skin rash, they're not maintained on this therapy. So we can see response markers of efficacy and safety. Now, perhaps most compellingly of all, when we look at pembrolizumab, this was the gold standard anti-PD-1 immune-enhancing or immune-recovering antibody in the tumor microenvironment models. Outside the tumor microenvironment, this is absolutely critically not inflammatory. And this is fundamental. This is the difference between the success of the anti-PD-1s and the catastrophic failures of the anti-CD28 T-Gennaro compound over a decade ago. So this is a way in which we can use these two panels to evaluate the impact on the immune in the selective microenvironment, tumor microenvironment, and in the patient as a whole. So finally, I just want to end by showing you how BioMAP systems have been very, very valuable and compelling to clients evaluating combination therapy. This, of course, is really not amenable to many in vitro approaches. Current animal models have been shown to be poorly predictive as well as being expensive for a combination trial. And often this is really just reported or even anecdotal data that comes out from the clinic. So here we have built a special mathematical template in which the agents are arrayed singly as individual agents. Here we've agent B and agent A, and then we've built this matrix of dosing that enables us to test 16 samples of the 4 combinations of each in triplicate. And just to really illustrate, here we have an overlay of Paclitaxel, which had immune recovering activities in both the stromal system and the vascular system, as did pembrolizumab or Keytruda, the anti-PD-1. And at concentrations here represented in the combination that was tested in parallel, we actually see an enhanced immune recovery. It's not going to be huge because it can't be. Really it's not driving inflammation in these systems, it's recovering it. But what we see is a compelling gain or enhanced or even potentiated immune response in the stromal system, and particularly noted in the vascular system. So just to summarize what I've shown you today, we have identified assays and services that we have at DiscoverX that really can help provide valuable information for you at each of these stages of the drug discovery continuum. The PathHunter assays can be used to rapidly identify, validate, and optimize candidate molecules. The BioMAP systems then can be used to inform your compound suitability for IONC. Does it have efficacy-related activities? Are these immune recovering activities? And then for safety, what's my compound or combination doing outside the tumor microenvironment? That can be tested as well. And finally, is my combination relevant? Is my combination statistically significantly different from both agents alone? If it's not, it may not merit a large clinical trial. So these data are designed to help inform and guide next steps in progressing compounds and combinations, not only along the discovery continuum, but into the market and into the clinic. So with that, I will hand over to Sega, and we're happy to take questions for you, and I thank you for your attention.
Okay, great. Thank you very much, Alison. It's always very educational for me, from on a personal note, and always a pleasure and a treat for me. So at this time, we will be taking any questions that you may have. please let us know. And right now we'll be dropping audio for just a moment. Okay, thank you very much. We received our first question. Are your BioMAP oncology systems able to test small molecule and antibody combinations?
Yes, in fact, the last data slide I showed demonstrated the gain of efficacy or enhanced efficacy we saw when we combined the small molecule Paclitaxel with the large molecule anti-PD-1 pembrolizumab. And so, in fact, yes, very much so. We can see or we can utilize BioMAP systems for testing combinations of small plus large molecules.
We received another question from one of our participants, and it's very difficult sometimes to purchase clinical grade antibodies, especially in Asia. Is it possible for DiscoverX to purchase these clinical grade antibodies to test in BioMAP oncology systems?
Yes, provided they are available on a pharmacy grade. Our pembrolizumab and nivolumab were indeed pharmacy formulations that we tested in our system. We do have a partnership with not a partnership, but we do have access to a company that can procure those agents if needed.
Great. So we received another question. What advantages does the BioMAP oncology systems have over, let's say, MLR, mixed lymphocyte response?
That's a terrific question, an important question, and there's actually a couple of answers if I can address them one by one. So there's almost the logistical answer that MLR will require select donors, and then you worry about your donor variability from experiment to experiment, and then the logistics of it being a prolonged assay. So if you take donor 1 and donor 2, will it be the same as donor 3 and donor 4? And so BioMAP certainly has been developed and optimized and validated to be more reproducible for every donor pool, even though they're not the same donor pools, but they are more reproducible. And I think the second, more biological or clinically relevant difference between BioMAP systems and MLR is that we are building in the complexity of the tumor tissue. So again, as I said during my presentation, immuno-oncology is built, or immuno-oncology therapies are built on the premise of tolerance, of the exploitation of tolerance mechanisms by the tumor. And so these agents have to work in a complex environment with which normal tolerance must be addressed also. And so when you come in with, particularly with a large molecule, you're going to be competing with the tissue and the immune cells and the tumor cells for your target and your target biology. And so knowing the cumulative phenotypic impact of that can be helpful. So two things, logistics and then biology.
Okay, another question that we received, this is probably related more towards the Diversity Plus system. You know, being that you mentioned that a lot of the PBMCs have been pooled from at least 3, maybe up to 5 different patients. In your BioMAP systems, especially in Diversity Plus, how do you manage donor variability between each of those systems?
We manage donor variability in 3 ways. The first is that we pool and stimulate the systems. So it turns out that donor variability is in fact very real, but often is the delta between the basal and the stim is much more uniform between individuals. So while your basal TNF might be 1 and stimulated might be 10, mine might be 10 and 100, and the delta for both of those would be 10. And in fact, that's the basis of how we diagnose a disease, right? The disease or stimulated phenotype is more consistent across individuals. So stimulating the system mitigates a lot of the donor-donor variation because we're looking at the elevated or tissue and disease state. The second way we manage it is that the data is expressed as a delta to the log, or excuse me, a log ratio of the delta between the compound treated and the control treated wells. And the biology and the cells and the donors are exactly the same between the vehicle and the compound. So really all you're seeing on the graph is what was the delta caused by my compound on the biology. And finally, by using pools of donors, you can equilibrate the variation you see from individual donors. So that's the 3, pools, stimulation, and the delta with the vehicle control.
Okay, we received one more question. Have you tested your compounds in co-culture using, rather than healthy donors, patient-derived PBMCs?
Actually, this is really more not just relevant to immuno-oncology. This is relevant to all of our systems. We get asked this question for our autoimmune models, for our fibrosis models. One of the challenging things with using cancer patients or using any patient-derived cell types is that they come not only with the disease heterogeneity on board, but they often come with significant drug pressure on board. So using PBMCs from patients limits screening technology, and you also require a significant history of the drug treatment. An example of this that we came across was actually in a system that was relevant for lupus, where we wanted to compare our BT system to a BT system with PBMCs from lupus patients. And after extensive, more than a year and a half of studies, what we found was that the variability in the patients was enormous. We can only use individual patients. But when they present at the clinic, at the time that you get the PBMCs, is usually because they've been having a flare-up, and they may often have self-medicated. So the drug pressure of patient-treated cells is really almost an unfair approach to screening campaigns. How do you understand the impact of a compound on a drug-pressured system? So what we've done instead is we've taken a healthy, drug-free cell population and driven it into a disease state, and then allow that to assess the impact of the compound. All of the systems are validated with clinical standards of care, and so we optimize them to capture the clinical response biomarkers, and then that allows us to test compounds in a drug-free background.
Okay, wonderful. There are a couple more questions that we've received, but due to some time constraints, we are going to conclude our webinar today. Again, thank you very much for attending. I know it's first thing Friday morning, so thank you very much. Hopefully Allison and I will go out for a glass of wine after this. But Allison, thank you again. Really appreciate your time.
I hope that maybe some of the people, we can get there and visit in person sometime.
That would be wonderful. That would be wonderful. Again, thank you very much, everybody, for attending. We will be concluding our webinar. Slides will be sent to you immediately following this presentation, and once we can get the streaming video, we will also be sending that link to you via email, the email that you registered for this webinar with. Again, thank you very much, and thank you, Allison, for your time.
You're welcome. Thank you. Bye-bye.